Hello class, I hope everyone is doing well at home. My name is Thea Javria, and today I will be explaining to you guys macrophage polarization therapy on asthma patients. So first, I'm going to go over our student learning outcomes. Upon completion of this presentation, students should be able to understand what asthma is and its effects on the body, be able to identify key concepts of macrophages, understand macrophages' role with asthma, and understand the key concepts of macrophage polarization therapy. So let me explain a little bit of what asthma is. Asthma is an inflammatory condition in which substances from the environment will induce narrowing and swelling of the airways. You can see this on the diagram on the right side of the slide that there is a normal to severe asthmatic airway. The normal one is open and allows air to flow through it, while the one on the right you can see is inflamed and swollen basically all the way. And the opening that it does have, it is covered in mucus that the lungs have just produced to protect itself. On the left side of the slide, you can see there's a diagram of the immune response that happens when a foreign object, or in this case, the antigen, comes in contact with the lungs, which causes this cascade that ultimately leads to inflammation and asthma symptoms. I'll go deeper into detail about this diagram later. There are currently 3 classifications of asthma based on how it is induced. There's exercise, occupational, like through chemical fumes or gases you could find in the workplace, and allergy-induced. Seeing the airway that it has, you can kind of assume that the symptoms of asthma would include shortness of breath, tightness of the chest, wheezing, trouble sleeping, coughing, or wheezing attacks. I'm going to explain a little bit why this is important. Asthma currently affects every 1 in 12 people, and those numbers increase in value every year. It presents itself differently in each person, but it does continue to be a lifelong condition that does not have a cure, but it is treatable. People often use medication to help handle their flare-ups and to counter their asthma, and the best and most common treatment now is the use of glucocorticoids, which have an anti-inflammatory effect that bring down the swelling of the airways in the lungs. Studies have shown, though, that people have had a resistance to this type of drug, and there have been other instances where regular treatment is ineffective. Which brings me to why there is a need to find other treatments like macrophage polarization therapy. Macrophages are a large part of our innate immune system and are especially present in the lungs, where they are the most abundant and make up about 70% of the immune cells present. They are a large phagocytic cell that is used to consume particles such as bacteria, and also can activate different responses like inflammation, which is what we're going to focus on for this presentation. Their functions include keeping homeostasis and development within tissues, tissue repair, and most of all, immunity. That is why they are such a target for therapeutic research, because many diseases are associated with chronic inflammation like cancer, arthritis, diabetes, and what we're going to be focusing on today, asthma. Studies have shown that macrophages have different receptors on their surface that respond to different stimuli from the environment that lead to different pathways. The research I read focused on getting a better picture of what kind of cell signaling pathways are triggered when confronted by a controlled stimulus, in hopes that we may be able to one day alter certain pathways and cause less or more of a response. So in this diagram that I'm showing here, this is the pathogenesis of asthma. You'll see that an antigen is being introduced, and it will come across a naive T lymphocyte, which will differentiate into either a T helper type 1 or T helper type 2. Th1 or Th2 for short. These T helpers will signal towards other immune cells to target that antigen to cause a response like inflammation. Th2 is largely associated with allergic asthma because, as you can see here, it produces a much larger immune response, which brings about an increase in inflammation. And produce inflammation-inducing substances like histamine. Through experimentation with different allergic and non-allergic samples, they were able to identify M1 and M2 macrophages and their association with different pathways. So in the experiment that they had in my research paper, they were trying to see how M1 and M2 macrophages fit in these pathways, and they took FDE, farm dust extract, which induces a non-allergic response associated with Th1, and HDM, home dust model, which induces an allergic response associated with Th2 and exposed them to mice. It was a 6-week experiment with eight mice each for HDM, the FDE, and a control, and they were able to tell which macrophages type belonged by comparing it to the amount of cytokines produced at the end. So at the end, the experiment, they aestheticized the mice, euthanized them, and harvested their lungs. They ground it up, and then they were able to kind of see um, what was produced in them. So the results show that the FDM are non-allergic stimulus induced M1 and not M2 by the amount of cytokines that it produced, and while HDM are allergic stimulus induced M2. So in our pathway, Th1 would release IFNY, interferon Y, which would activate M1, and Th2 would release a variety of interleukins that would activate M2 and cause asthma inflammation. So these are the areas where therapy research would be targeting to get our correct response. There are different ways that they're trying to research to target these macrophages. It is not yet an exact science. This is still fairly new, but they're trying to create drugs that target the surface molecules on macrophages. So it's trying to target the certain receptors that are on the surface of those macrophages. And another way is that they're trying to disrupt signaling pathways in tissues like the lungs. The example that they gave in the paper was kind of just like if there was a known disease that caused interferons as a stressor of that disease, then the macrophages would interfere and stop that interferon. The most research has gone into targeting epigenetic and transcriptional levels to change the signaling pathways of macrophages. So in the research, they mostly focused on 3 main mechanisms. Those mechanisms being miRNAs, DNA methylation, and histone modification. So in miRNAs are non-coding RNAs, and they regulate gene expression by reducing translation of target mRNAs that are regulators for Th2 activation. Studies are showing that miRNAs have the ability to balance M1 and M2 macrophage responses through transcription factors. There is also DNA methylation, which is the process of adding methyl groups to DNA molecules. Specifically, it uses DNA methyltransferases, or DNMT for short, which leads to the prevention of transcription. The process's name itself is gene silencing because the aim is to suppress that gene expression, and for this case specifically to target M2 macrophages that are known to cause asthmatic inflammation response. And lastly, there is the histone modification, where they use histone acetyltransferases, or HATs for short, and histone deacetylases, HDACs for short, which are associated with transcription expression and repression. This study shows that HATs and HDACs imbalances could be the cause of different diseases like asthma. HDAC resistance has actually been associated with steroid resistance, like with the use of inhalers, and the severity of asthma. So there's still a couple of issues that are being studied, like how stable would programming these cells be, the amount of time cells would be able to be used, is this a long-term solution or is it another short-term? A large portion of the studies were done in vitro, meaning most of these studies were not done with a living organism, which can present problems later. Macrophage polarization therapy is a promising study that could bring answers to many questions about the body and different diseases. Like I said before, this is all still fairly new, and the study with the mice was even done in like 2015, so which is not that far away. And with new discoveries every day, it is possible that we can make a lot of advances and better our understanding of the many signaling pathways of our body. I know for myself that I have asthma, and I've had asthma since I was a kid, and that I would love a long-term solution because I feel awful especially this time of year where it's like springtime and there's pollen everywhere and I just have a lot of trouble breathing. You might be able to hear it in my presentation, but if there is a long-term solution, I would really appreciate it. All right, and here are my references. Thank you.